Let's get started. My name is Jerry Walter, and I'm the founder and president of the National Fabry Disease Foundation. Thank you all for being here tonight. Well, we have Dr. Chris Hopkins talking about Glafabra. So what's going to happen next is I'm going to introduce you to Casey McKenna, who is the NFDF Senior Program Manager for Disease Education Awareness and Community Assistance Programs. And Casey is going to introduce you to Dr. Hopkins. Casey?
Great. Thank you, Jerry. As Jerry said, we are pleased today to have Dr. Chris Hopkins with us today. Dr. Hopkins holds a PhD in biochemistry, cell, and molecular biology from Boston University. As an entrepreneur, Chris leverages his PhD and MBA to develop advanced therapies to treat various genetic diseases. Currently, as CEO of Glafabra, Chris leads a team of researchers skilled in cell therapy development and drug commercialization. Glafabra is investigating a cell-based therapy to treat Fabry, Pompe, and Gaucher diseases with a treatment with a longer half-life than enzyme replacement therapy. But Chris is going to tell us quite a bit more about that. If you have questions that come up along the way, feel free to pop them in the chat, and I will ask them if we need to make sure we are on the same page before we're moving on. Otherwise, we'll take questions at the end of Chris's presentation.
And thank you, Dr. Hopkins. And if you would like to share your screen.
All righty, will do. Okay, hopefully you can start seeing that.
Yep, we can see it.
All right. Well, thanks everyone for being here today. I really appreciate you and let me speak to you. Yeah, we're a startup, a little bit of background. We're brand new at this, but we're actually taking something that another company did. You may have heard of AVROBIO. They more or less shelved the asset, and we're basically trying to revive that asset. We've rebranded it, and we're calling it the live cell therapy. And so that therapy has been in the clinic for Fabry disease, and I'll show you some of the data that we have for that. But basically, it's a long-acting therapy. And we found that just one dose can last for years, not weeks. Now, I presume most everybody knows about Fabry disease, but I'm going to go ahead and sort of, put us all on one page, with a little bit of an intro and use this guy here. This is Joe. Joe has Fabry disease. He is struggling with the buildup of toxic lipid metabolites in his body because he's missing a key enzyme. Now, thankfully, Joe has access to the standard of care, which is ERT. Joe knows he needs ERT, but he doesn't like it. He needs to go into the clinic and get those enzyme replacement therapy. And with that, we're calling that getting the jab for the IV bag every other week. And this is challenging for Joe. What if we could offer Joe a new therapy, one that lasted for years and not just a few weeks? Well, we have that solution. It's our live cell therapy. Joe can take a one-time dose and get 5 years of relief. So imagine Joe getting back all that time with his family and being able to live a more normal life. Now, how does our therapy work for Joe? Well, we harvest the cells out of Joe, his immune cells. We then modify these cells so that they will make the missing enzyme. We then return those cells back into Joe as a one-time infusion that now allows those cells to create progeny that make white blood cells that go everywhere in the body and start to secrete the missing enzyme. Now, in a process called cross-correction, the neighboring cells can take up that secreted enzyme and use it to stop the buildup of toxic metabolites. Joe gets to take this therapy as a one-time dose and gets years of relief. Now, it's that durability that is a key patient value. So with that standard of care being the enzyme replacement therapy, or T, patients need to get that enzyme infusion, and they need multiple treatments every other week for the rest of their lives to control their Fabry. Contrast that to our live cell therapy, and Joe's going to be able to take a one-time cell infusion. It's going to provide continuous levels of enzyme in his body, and that level of enzyme is going to be maintained and last for years. Now, our live cell therapy has been tested in patients in a Canadian clinical trial on 5 patients. Joe was one of our first volunteers. We measured the toxic metabolites in Joe and his clinical cohort to determine the effectiveness of our therapy. Initially, all patients were brought into the study and were on ERT. When we took them off ERT, we saw a 41% jump in the level of toxic metabolites, lyso-Gb3. Then when we started them on our cell therapy, we saw a 48% drop. And the signal there for the 5 patients is actually lower than the signal they were getting when they were having ERT. And what's interesting is 5 years later, we went back to the patients, reanalyzed them to look at their toxic metabolites, and found that they still were in good control. And that's 5 years after that first initial dose. I have a quote from Joe, and actually, it's another member of your community, but I'm changing the name to protect the innocent. Joe did say that the benefits were many after his therapy, and that he's felt the best he has felt in his life. Data for 5 patients is shown here, and it's basically that same plot we just saw, but now we're doing a little bit of normalization, and we're looking at every individual's data point here. So initially, they were normalized in the very beginning. And then when measured again for their ERT, we're seeing this little bit of scatter of response between each of the patients. But again, normalized to 0, we didn't see a huge amount of jump. Then when we took them off ERT, just about every patient saw a good sized jump. Maybe the black one here didn't show too much. But then when we started them on the cell therapy, we saw a significant drop. There's a bit of a caveat here is that after being started on cell therapy, the protocol called for them to, all, if they wanted to go back on ERT therapy. One of the patients said, no, I don't want to go back on ERT. 4 of the 5 patients said, let's go back on ERT. And then after a couple more months, it was recognized that the levels of toxic metabolites were low enough that they could be elected to go off ERT. And so of the 4 patients, 2 of them elected to go off. So 2 in this have maintained their enzyme replacement therapy throughout. And so I'm just trying to get that window out of the way. And so that's the ones that maintained it throughout were the ampersand one, the black line. They left their ERT and then they never came back to the ERT and saw a better response with the cell therapy. And then the pound sign is the ones that went starting back on at 3 months back on ERT and never left it. So that's here, the orange one and the blue one. And then the remaining 2 had gone off their ERT after 3 to 12 months. It turns out that this patient in yellow appeared to have sort of lost the engraftment and needed to go back on ERT at year 4. So 4 out of 5 patients at year 5 are still going strong without needing to do ERT, but one of those patients did need to go back on ERT. And so that's sort of the detail of the data. Let's look at some of the safety data. Here's 2 different sort of measures. The top line is for essentially measuring kidney parameters, and the bottom line is for measuring heart parameters, so kidney or renal. The urine protein, proteinuria, there was the blue patient in the blue showed a bit of a noise and then kind of stabilized in the proteinuria, the urine protein, proteinuria. And then the estimated glomerular filtration rate, they had a fairly low estimated glomerular filtration rate, but they ended up stabilizing. Turns out this person barely made it into the study because that person in the blue was already showing clinical signs of kidney failure. For the troponin and then the left ventricular mass index, we're seeing that patient 1 was maybe a little bit elevated, but still showing, not showing an escalating effect for either of these 2 cardiac parameters. So it looks like both renal and cardiac outcomes are not dramatically altered by the therapy. The durability is somewhat measured in looking at the amount of alpha galactosidase in both plasma and leukocytes over time. And it's also can be sort of seen in the vector copy number when you go into your leukocytes and ask how many of those are containing the transgene, the alpha galactosidase, the gene coding for alpha galactosidase. So when we first look at this, we see that, before time 0, we're at a very low level. There's a little strangeness going on in our kidney patient, but for the most part, he jumps up, and then kind of maintains a steady state level of alpha galactosidase. Some of these others are showing a really good jump. The patient number 5 jumps up and stays in plasma at a very normal level. That's the 2 dotted lines here. Leukocytes, red's actually way above normal in the leukocytes, and then a lot of the other patients were showing some pretty good levels of alpha galactosidase in their leukocytes. And the vector copy number again spikes up and sort of fizzles, sort of decays down. And some reach sort of a steady state level, and others are still sort of decaying a little bit. So the perdurance of this therapy, how long it lasts, might, we're looking at 4 to 5 years, maybe even a little bit longer, might be the average length that a dose can last in a patient. And so we have some pretty good levels of transgene expression in all the patients for at least up to 4 years. The trials were started in 2016, and then the 5-year follow-up was completed in 2023. We saw a lot of peer-reviewed publications were achieved in that time frame, and the key ones were able to demonstrate the therapy is safe, effective, and durable. Now, Joe liked his live cell therapy because it gave him lifestyle freedom. Joe saw that long duration, 5 years from just that one dose. Joe also saw a large effect, allowing him to be able to say that he's felt the best he's felt in his life. Now, Joe has some alternatives. Joe could go take AAV therapy. That's one of the other emerging therapies that stands for adeno-associated virus. AAV therapy is like us. It has a long duration and a large effect. But the challenge with AAV is in its current forms, it's not repeatable. So when these patients start to fizzle out after a few years, as has been happening in some of those clinical trials, they will need to seek an alternative option, and that could potentially be us, the live cell therapy. Now, Joe can say that he's felt the best he's felt in his life because our therapy has the capability of reaching nearly all tissue, including getting across the blood-brain barrier and treating some of those CNS symptoms. And there's some anecdotal evidence or suggestions that that may be occurring. Think about ERT and AAV, very big difficulty getting across the blood-brain barrier. And then finally, we're planning to price our therapy at a fairly affordable price. The ERT therapy, depending on, even for a 5-year cost, that's actually kind of a low number. But anywhere from 2 to 3 million is the 5-year cost potentially, depending on the person. We're planning to be able to price our therapy close to what is CAR T therapy. So CAR T therapy is a therapy that is a treatment for cancer, and it's a cell-based treatment. But basically, we're utilizing the very similar manufacturing process that is used to make CAR T therapy. And instead of creating a chimeric antigen to attack cancer, we're instructing those cells to make and secrete the missing enzyme. And so that very pattern, or the what's called the CMC, the chemistry manufacturing, basically the production of the therapy is well worked out for CAR T, and we can essentially jump in there and utilize the exact same manufacturing system. So we know we should be able to price this at a nice affordable price of about $500,000 per dose. And it's going to be more cost effective than the dose that you see with the current standard of care. We're going to be doing clinical trials, hopefully very soon. We're still trying to get the funding to get us there. But what we want to do is to obtain an IND with the FDA. And the steps we're going to take is to get an orphan drug designation, then get an interact meeting with the FDA so that we can get clear guidance on what we need to do to be able to repeat the Canadian trials in the US. We then, part of that's a pre-IND meeting and submitting the IND and getting FDA clearance and getting an open IND. And this will allow us to essentially activate our main clinical site, which we've already have an LOI signed with the University of Utah to be our site, headed by Dr. Brian Shioda. And so we're also trying to select some additional sites that potentially can be satellite sites to help both provide patients and monitor the patients after the therapy. The clinical roadmap here, I took a sort of a Google Maps kind of thing. We're first going to be starting off getting, taking a turn at the orphan drug designation. Next turn is going to be at the FDA Interact meeting. And you can see here then the little blue line, we're sort of going to go through a bunch of different steps before we essentially get into trial. And it's a long kind of, it's still a couple multi-year process to get to market approval. So just trying to set the expectations there. We are also planning to create treatments in other lysosomal storage disorders, other enzyme deficiencies. Our two key ones we're sort of holding up right now as seeming like the ones that we think we can do make a big difference is in Pompe and Gaucher. but we could also potentially treat a wide variety. In fact, there's 48 other lysosomal storage disorders. And in fact, even all enzyme deficiency disorders potentially could be treated by our therapy with some of the tweaks in the secretory apparatus. The team we have is myself, Chris Hopkins, is CEO. I started a company called Nutratransgenics in 2010, grew it to the point where I was able to get it exited in Vivo Biosystems. We have Elizabeth Wagner as on our team as the COO. She's had some experience with some exits. She also has worked in Genzyme and was working on the Fabrazyme development before it was sold to Sanofi. Krista Casaza helps us in regulatory science, and the two co-founders who've been amazing in the space and been working at it for 30 years, Dr. Jeffrey Medina and Ronan Foley, who are my co-founders and partners in this effort. But we also have some other business advisors and some of the science advisors you might recognize who are helping us shore up gaps and help us give it a strong team to help see success. Um, to summarize, we're bringing live cell technology to the market. We're creating therapies in Fabry, Pompe, and Gaucher. Our Fabry asset is already been in the clinic and Canadian trials in Canada. It's been demonstrated to be safe, effective, and durable. We're going to end on one more quote from Joe. At one point, my wife and I realized we were forgetting I had a Fabry at all. And with that, I can take questions.
So thank you very much to Chris, and I'll hand it back over to Jerry.
Thank you, Dr. Hopkins, and it's exciting to have another potential solution for people with Fabry disease. And thank you to Casey, and thank you to all of you for being with us tonight. And with that, we'll say good night.
I will say that anybody feels like reaching out to me, please do. I would love to try and answer all these questions. We got to get answers for this stuff. So reach out and I'll figure out how to get them done.
Great.
Great.
And I don't know if you can see it, Chris, you're getting lots of applause and thank yous in the chats.
Thank you, everybody.